How did almost every animal body plan appear in just 25 million years? In our last episode, you witnessed the Precambrian Stage 3 billion years of preparation that built the foundation for complex life. You met the Ediacaran organisms like Dickinsonia and Charnia, peaceful creatures perfectly adapted to a world without predators. But that peaceful world was ending. Now, 541 million years ago, we stand at the threshold of the most dramatic transformation in the history of life. This episode reveals what happened when evolution shifted into overdrive, when predators emerged, when shells and eyes appeared, when nearly every major animal body plan exploded into existence within just 10 to 25 million years. This is the Cambrian Explosion, and you're about to witness life going wild. Here's a fact that challenges everything we think about evolution. It took 3 billion years for complex animals to appear, but only 10 to 25 million years for virtually every major animal body plan to emerge. Think about that time frame. 25 million years sounds long, but in geological terms it's a blink less than 0.5% of Earth's history. Yet during this brief window, oceans transformed from simple gardens of soft-bodied organisms into complex ecosystems filled with predators and prey, armored defenders and sophisticated hunters. Arthropods appeared, ancestors of modern insects, spiders, and crustaceans. Mollusks emerged, relatives of today's snails, clams, and octopuses. Echinoderms, brachiopods, annelid worms, and chordates, the group that includes Usol, appeared during this explosion. What triggered this sudden burst of innovation? Why did evolution, which had been patient for billions of years, suddenly accelerate? The answer lies in a single, revolutionary change: predation. For the first time in Earth's history, animals began eating other animals. And when that happened, everything changed. The peaceful garden became a battlefield, and the arms race began. You're about to meet the pioneers of this new world, creatures with eyes, jaws, armor, and weapons. Some will look familiar, their body plans still successful today. Others will look utterly alien, evolutionary experiments that worked brilliantly in their time but left no descendants. This is the moment when life exploded. This is when the modern world began. Evolution, the triggers that sparked the explosion. The Cambrian Explosion didn't happen randomly. It required specific conditions and triggers that all converged at the same moment in Earth's history. What were those triggers, and why did they matter? Trigger 1. The evolution of predation. The most important trigger was predation itself. Before the Cambrian, feeding was passive. Organisms absorbed nutrients, filtered particles, or grazed on bacterial mats. But then something revolutionary happened. Some organisms developed the ability to actively hunt and consume other organisms. How did predation evolve? Gradually. Perhaps it began with organisms accidentally ingesting smaller creatures while or maybe some creatures started scraping not just bacteria but also the soft tissues of other organisms. If you're being hunted, you need defenses, shells, spines, speed, or the ability to burrow and hide. If you're a hunter, you need weapons, jaws, claws, improved senses, and mobility. Suddenly, evolution isn't just about growing bigger or absorbing nutrients more efficiently. It's about survival in a dangerous world. The arms race begins and innovation accelerates. Trigger 2. rising oxygen levels. Oxygen continued to rise throughout the late Precambrian and into the Cambrian, reaching levels between 15 to 25% of the atmosphere, approaching modern concentrations of 21%. Why does this matter? Because predation and active lifestyles require enormous amounts of energy. Chasing prey, fleeing predators, building armor, powering complex nervous systems, all of this demands the energy budget that only oxygen-based metabolism can provide. Higher oxygen levels made active, complex lifestyles not just possible but advantageous. Organisms that could exploit this energy windfall outcompeted those that couldn't. Trigger 3. Environmental stability and habitat expansion. The early Cambrian provided stable, favorable conditions. Sea levels were high, creating vast, shallow marine habitats across continental shelves, perfect environments for diversification. Temperatures were moderate, ocean chemistry was favorable, and nutrients were abundant. These conditions created ecological opportunities. Empty niches existed everywhere. No one had figured out the best way to be a predator, a grazer, a filter feeder, or a burrower. Evolution experimented with every possibility, filling these niches with wild diversity. Trigger 4. Genetic innovations. The genetic toolkit for building complex bodies, Hox genes, and other regulatory systems was already in place by the Cambrian. But the Cambrian saw something new: gene duplication and regulatory network expansion. When genes duplicate, one copy can maintain the original function while the other mutates and tries new variations. This creates raw material for innovation without losing essential functions. During the Cambrian, gene duplication events allowed organisms to develop new features: eyes, limbs, segmented bodies, while keeping their basic systems intact. The perfect storm, all four triggers converged simultaneously. Predation creating selection pressure, oxygen providing energy, environment offering opportunities, and genetics supplying the tools. The result was explosive diversification, the Cambrian Explosion. Anatomy: the diverse bodies of Cambrian life. The Cambrian saw an explosion of body plan diversity that has never been matched. Let's examine 5 iconic creatures in detail, exploring both external and internal anatomy. Trilobites, masters of armored complexity. External anatomy. Trilobites had bodies divided into 3 longitudinal lobes, a central axis flanked by 2 lateral lobes and 3 main body sections. The head shield, cephalon, segmented thorax, and tail shield, pygidium. The head shield was remarkable. 2 compound eyes sat on the upper surface, each composed of calcite lenses, transparent calcium carbonate, forming precise optical structures. Some species had eyes with thousands of lenses, providing nearly panoramic vision. Between the eyes, a raised central area called the glabella protected the brain and digestive system. Long genal spines often extended backward from the head corners, possibly for defense or stability. The thorax consisted of multiple segments, each bearing a pair of jointed legs underneath. Species varied from a few segments to dozens, depending on lifestyle. The segments were articulated, allowing many trilobites to roll into a ball-complete protection against predators. The tail shield provided a defensive rearguard, and in some species, additional spines or ornamental structures. Trilobite exoskeletons were composed of calcite, making them incredibly hard and fossilization-friendly, which is why trilobites are among the most common Paleozoic fossils. Internal anatomy. Beneath the armor, trilobites had surprisingly sophisticated internal systems. A simple but functional digestive tract ran from the mouth underneath the head through the body to an anus near the tail. The gut was straight or slightly coiled, processing detritus, small organisms or organic matter. A basic circulatory system pumped blood, hemolymph, through the body cavity, though trilobites lacked true blood vessels. The hemolymph carried nutrients and oxygen absorbed through the legs and body surface. The nervous system consisted of a brain cerebral ganglion in the head, connected to a ventral nerve cord running the body length. Each segment had ganglia controlling local functions. Decentralized system allowing coordinated movement even if part of the body was damaged. Trilobites breathed through gills attached to their legs, thin feathery structures absorbing dissolved oxygen from water as legs moved for walking or swimming. Size and variation. Most trilobites were 3 to 10 centimeters long, but some giants reached 70 centimeters. They came in incredible variety: flat species for gliding over mud, spiny species for defense, streamlined species for swimming. This diversity made them one of the most successful animal groups ever. Anomalocaris, the first apex predator. External anatomy. Anomalocaris was massive by Cambrian standards, up to 1 meter long. Its body was segmented and laterally compressed, with flexible, overlapping lobes running down each side. These lobes undulated rhythmically, creating wave-like propulsion that pushed the animal forward, an efficient swimming method, allowing sustained speed. The head was dominated by 2 enormous compound eyes on stalks, each containing up to 16,000 individual lenses, the most advanced eyes of the early Cambrian. These eyes provided exceptional resolution and motion detection across a wide visual field. The most distinctive features were the 2 large grasping appendages extending forward from beneath the head. Each appendage was segmented, flexible, and lined with sharp spines along its inner edge. These arms could reach out, close around prey like grasping fingers, and pull victims toward the mouth. The mouth was circular, located centrally on the underside of the head. It consisted of overlapping plates arranged like a pineapple ring, capable of constricting and expanding to manipulate prey. The plates had ridges that could crush soft tissue or hold struggling organisms. A fan-like tail at the rear provided steering and additional thrust during hunting pursuits. Internal anatomy. Anomalocaris had a relatively simple but effective digestive system. Straight gut running from the mouth through the body to a posterior anus. The gut was muscular, capable of peristaltic contractions to move food through digestion. The circulatory system was open, with hemolymph flowing through body cavities rather than enclosed vessels. A dorsal vessel acted as a primitive heart, pumping hemolymph forward, then letting it percolate through tissues before returning. The nervous system was sophisticated for its time. A large brain in the head processed visual information from those enormous eyes, coordinated grasping appendage movements, and controlled swimming patterns. A ventral nerve cord extended through the body with segmental ganglia. Respiration likely occurred through the body's surface and specialized gills along the lateral lobes, absorbing oxygen as water flowed past during swimming. Opabenia, the five-eyed enigma. External anatomy. Opabenia was small, about 7 centimeters long, with a soft, segmented body bearing lateral lobes for swimming, similar to, but smaller than, its most bizarre feature: 5 eyes arranged across the top of the head. 3 eyes formed a forward-facing triangle with 2 additional eyes positioned behind and to the sides. Each eye was stalked, projecting slightly above the head surface. This arrangement provided nearly complete spherical vision. Opabenia could see in almost every direction simultaneously. The feeding appendage was extraordinary: a long, flexible, segmented proboscis extending from the front of the head, lined with spines. At the tip was a claw-like, grasping structure. This proboscis could extend downward to the seafloor, grasp small prey with the terminal claw, then curl backward to bring food to the mouth located underneath the body. The tail was fan-shaped, providing propulsion and steering. Internal anatomy. Opabenia had a simple digestive tract from the ventral mouth through the body. The gut was short and relatively undifferentiated, suggesting it fed on small, easy-to-digest prey like worms or soft larvae. The nervous system was more complex than body size might suggest. Processing information from 5 eyes required substantial neural capacity. The brain had distinct optical lobes for each eye, integrating multiple visual fields into coherent spatial awareness. Respiration occurred through the body surface and possibly through specialized areas along the lateral lobes. The small size meant diffusion alone could meet oxygen demands without dedicated respiratory organs. Hallucigenia, the walking spine fortress. External anatomy. Hallucigenia was tiny, 1 to 2 centimeters long, with a worm-like body bearing 7 pairs of long, stiff, defensive spines running along its back. Each spine was rigid, pointed, and extended upward, creating a formidable barrier against predators attempting to bite or swallow it. Underneath, 7 pairs of stubby, jointed legs ended in small claws. These legs weren't for fast locomotion, but for anchoring the animal to surfaces, cephalour, sponges, or algae while it fed. The head was bulbous, with a simple mouth opening, possibly surrounded by small sensory papillae for detecting food. The rear tapered to a simple tail. The entire body had a wrinkled, segmented appearance with a tough cuticle providing additional protection beyond the spines. Internal anatomy. Hallucigenia had minimal internal complexity. A simple digestive tract ran from the mouth through the body, processing organic detritus, microbial films, or small organic particles scraped from surfaces. The nervous system consisted of a small brain and ventral nerve cord with segmental ganglia controlling the legs. There is no evidence of eyes or sophisticated sensory organs. Hallucigenia likely relied on chemical and tactile senses to locate food. Respiration occurred through the body surface. At such small size, diffusion alone sufficed for oxygen needs. Pikaia, our humble ancestor. External anatomy. Pikaia was small and streamlined, about 4 centimeters long, resembling a primitive eel or lancelet. The body was laterally compressed and ribbon-like, optimized for swimming through undulation. Running the entire length of the body was a notochorda, flexible rod providing internal support for muscle attachment. This was revolutionary. Instead of external armor or a rigid body cavity, Pikaia had internal structural support, allowing flexible, efficient swimming. Above the notochord ran a dorsal nerve called a holotube of nervous tissue that would eventually evolve into the spinal cord of vertebrates. Along each side, segmented muscle blocks, myomeres arranged in a zigzag pattern, contracted rhythmically to produce swimming undulations. The head had two small tentacles projecting forward, likely sensory structures for detecting food or water chemistry. No obvious eyes, no jaws, no defensive spines. A small tail fin at the rear provided final propulsion thrust. Internal anatomy. Picaya's internal anatomy was simple but contained the blueprint for all vertebrate life. The notochord provided structural support without weighing the animal down with armor. The dorsal nerve cord processed information and coordinated movement with surprising sophistication for such a small creature. A simple digestive tract ran ventrally below the notochord, processing small particles filtered from water or picked from the seafloor. Picaya likely fed on microorganisms, organic detritus, or algae. A simple circulatory system pumped blood forward through a ventral vessel, then backward through a dorsal vessel, with blood exchanging oxygen and nutrients through diffusion in tissues. Respiration occurred through pharyngeal slits openings in the throat, region that allowed water to pass through while filtering out food particles. These slits also absorbed oxygen, serving dual purposes of feeding and breathing. Why Picaya matters. Picaya wasn't successful by Cambrian standards. It was small, unarmored, and probably not abundant. But it represented the chordate body plan: notochord, dorsal nerve cord, segmented muscles, and pharyngeal slits. These features would prove extraordinarily versatile over the next 500 million years, eventually giving rise to fish, amphibians, reptiles, birds, mammals, and humans. Behavior, survival strategies in the Cambrian seas. How did these creatures actually live? The Cambrian introduced behaviors never seen before in Earth's oceans: predation and hunting. Anomalocaris was the Cambrian's top predator, and its hunting strategy was sophisticated. It patrolled the mid-water zone, scanning the seafloor below with those enormous eyes. When it detected perhaps a trilobite or soft-bodied wormit dove, extended its grasping arms, seized the victim, and brought it to its circular mouth. The mouth plates worked the prey, breaking it down enough to swallow. Evidence suggests Anomalocaris sometimes fed on trilobites, leaving characteristic bite marks, circular or crescent-shaped, damage matching the mouth structure. Some injured trilobites healed and survived, showing that not every attack was fatal, an early example of predator-prey dynamics we still see today. Opabenia hunted smaller prey. It swam slowly along the seafloor, scanning with 5 eyes for worms or tiny creatures hiding in sediment. When prey was located, Opabenia extended its proboscis, grasped the victim with the terminal claw, and curled the whole appendage backward to deliver food to the mouth. This feeding strategy allowed Opabenia to reach into crevices and spaces where larger predators couldn't access. Defense strategies. Trilobites developed multiple defensive strategies. The most dramatic was enrollment trolling into a ball with the hard cephalon and pygidium meeting, protecting the soft legs and underside. Many fossilized trilobites are found enrolled, suggesting this was an effective last resort defense. Other defenses included thick armor, defensive spines making them difficult to grasp or swallow, and camouflage, some trilobites may have matched seafloor colors or pattern. Speed was another option. Streamlined trilobites could swim rapidly when threatened, escaping to safer areas. Hallucigenia's defense was passive but effective. Spines. Any predator attempting to bite Hallucigenia got a mouthful of sharp spines, painful, potentially damaging, and certainly discouraging. This defense allowed Hallucigenia to feed openly without constant vigilance. Feeding and foraging. Trilobites were opportunistic feeders. Most species crawled across the seafloor, using their legs to stir sediment and capture small organisms, detritus, or organic particles. Some species may have scavenged dead animals. Others filtered particles from water using specialized leg structures. Picaia, likely filter-fed, swimming through water and capturing tiny organisms or particles in its pharyngeal slits. This passive feeding strategy required minimal energy but also provided limited nutrition, which explains why Picaia remained small. Burrowing and hiding. Many Cambrian organisms developed burrowing behaviors, digging into sediment to hide from predators or to find food. Trace fossils show complex burrow systems appearing during this time, evidence of organisms escaping the dangerous surface layer. Burrowing created new habitats and ecological niches. It also changed ocean chemistry. Organisms churning through sediment mixed oxygen deeper into the seafloor, affecting nutrient cycling and creating opportunities for other species. Social behavior, minimal but emerging. Most Cambrian organisms were solitary, but there's some evidence of aggregation, multiple individuals clustering in favorable areas. This wasn't cooperation or social organization, just independent organisms responding to the same resource concentrations. True social behavior, cooperation, communication, coordinated group activities would evolve much later. The Cambrian was too early for such complexity. Adaptation, meeting environmental challenges. The Cambrian wasn't uniformly favorable. Organisms faced multiple environmental challenges requiring rapid adaptation. Challenge 1. Oxygen fluctuations. While oxygen levels were generally higher than the Precambrian, they still fluctuated regionally and seasonally. Shallow waters might be oxygen-rich during the day when photosynthesis peaked, but depleted at night. Deeper waters often had lower oxygen. How did organisms adapt? Some developed more efficient respiratory systems, improved gills, enhanced blood circulation. Others tolerated wider oxygen ranges through physiological flexibility. Behavioral adaptations included migrating to oxygen-rich areas or becoming less active during low-oxygen periods. Challenge 2. Predation pressure. The emergence of predation was the greatest challenge. Organisms had to develop defenses quickly or face extinction. Shells and armor appeared rapidly across multiple lineages. Trilobites, brachiopods, mollusks, and others all independently evolved hard protective coverings. This convergent evolution suggests shells were the most effective defense available with existing genetic toolkits. Other adaptations included increased mobility for escape, burrowing to hide, growing spines or tough cuticles, and developing toxins or unpalatable tissues to discourage predators. Challenge 3. Competition for space and resources. As diversity exploded, organisms competed intensely for seafloor space, food sources, and favorable habitats. How did they adapt? specialization was key. Instead of competing directly, species evolved to exploit specific niches, particular food types, habitat depths, or microenvironments. Some trilobites specialized in sandy bottoms, others in muddy substrates. Some hunted active prey, others scavenged. This niche partitioning reduced direct competition and allowed coexistence. Challenge 4. Ocean chemistry changes. Calcium availability increased during the Cambrian, making shell building easier. But this also meant ocean pH and carbonate chemistry were changing, potentially stressing organisms adapted to previous conditions. Organisms adapted by evolving biochemical pathways for managing calcium carbonate precipitation, maintaining internal pH balance despite external changes, and tolerating wider ranges of chemical conditions. The result: rapid evolutionary innovation. These challenges drove adaptation at unprecedented speeds. The Cambrian shows evolution's capacity for rapid change when selection pressure is strong and genetic variation is available. In just 10 to 25 million years, life went from simple, soft-bodied forms to complex ecosystems with predators. Defenders, specialists, and generalists saw the ecological roles we see in modern oceans. Extinction and current status: who survived? Not every Cambrian body plan succeeded. Many bizarre experiments disappeared, while others became the foundations of modern animal groups. What determines success or failure? The Ediacaran holdovers: complete extinction. Any Ediacaran organisms that survived into the early Cambrian didn't last long. Their soft, passive body plans couldn't compete with armored, mobile, sensory-equipped Cambrian animals. By 530 million years ago, distinctive Ediacaran forms had vanished entirely. The Cambrian experiments: mixed results. Many unique Cambrian body plans also went extinct. The Burgess Shale preserves dozens of species that left no descendants. Sopabenia, Hallucigenia, though its relatives persisted, and many others. Why did these body plans fail? Often because they were too specialized for specific conditions. When environments changed, temperatures shifted, oxygen levels fluctuated, or new predators appeared, these specialists couldn't adapt quickly enough. Some were simply outcompeted by more efficient designs. Opabenius' five-eyed proboscis feeding strategy worked, but other predatory approaches proved more successful in the long term. The success stories: body plans that persist. Other Cambrian innovations were spectacularly successful. Trilobites dominated oceans for 270 million years before final extinction in the Permian mass extinction event. Their segmented, armored body plan proved incredibly versatile, adapting to countless niches. Arthropods, the broader group including trilobites, were even more successful. Modern insects, spiders, crustaceans, and other arthropods all descend from Cambrian arthropods. Today, arthropods represent over 80% of all animal species, making them the most successful body plan in history. Mollusks appeared in the Cambrian and thrived, giving rise to modern snails, clams, squids, and octopuses. Their shell-building capabilities and flexible body plan allowed adaptation to countless environments, from deep oceans to mountain streams. Chordates, like Pikaia, were humble Cambrian creatures, but their body plan internal support dorsal nerve cord proved extraordinarily versatile. From that small beginning came fish, amphibians, reptiles, dinosaurs, birds, mammals, and ultimately humans. Every vertebrate alive today descends from Cambrian chordates. What made the difference? Why did some body plans succeed while others failed? 3 factors. Versatility. Successful body plans could adapt to many environments and lifestyles. Arthropods could be predators, scavengers, filter feeders, or parasites. Chordates could swim, crawl, fly, or walk. Efficiency. Successful designs balanced competing demands: protection versus mobility, sensory sophistication versus energy cost, specialization versus flexibility. Evolvability. The most successful body plans had genetic architectures allowing continued innovation. They could generate variation without losing core functionality, permitting adaptation to changing conditions over hundreds of millions of years. Current status. The legacy lives on. The Cambrian Explosion ended around 485 to 490 million years ago, transitioning into the Ordovician period. But the innovations of the Cambrian persist in every animal alive today. When you see a butterfly, you're seeing Cambrian arthropod body plan innovation. When you watch a fish swim, you're seeing Cambrian chordate heritage. When you observe a snail, you're witnessing Cambrian mollusk success. The Cambrian established the blueprints. Everything since, including you, is variation on those ancient themes. The Cambrian Explosion transformed Earth from a planet of simple organisms to a world of complex ecosystems. It proved that evolution can generate extraordinary diversity in relatively brief periods when conditions are right. It also established patterns still visible today: predator-prey relationships driving evolutionary arms races, specialization allowing coexistence, complexity enabling new capabilities, and body plan versatility determining long-term success. In our next episode, we'll explore one Cambrian success story in depth: the Trilobites. We'll see how this group diversified into thousands of species, adapted to countless environments, and dominated Paleozoic oceans for 250 million years before finally going extinct. The explosion has happened. The blueprints are established. Now we'll see what those blueprints can build. The Cambrian proved that life, when challenged, doesn't just survive, explodes with innovation, diversity, and spectacular success. This is HistoPrimordia, where history comes alive. Thanks for watching. Subscribe now and join us for the next episode's story.